Thank you for standing by and welcome to the Arovella Therapeutics Investor Webinar and Q&A today, discussing the company's announcement this morning that it has licensed a novel solid tumour targeting technology. All participants are in a listen-only mode. There will be an initial slide presentation lasting for approximately 25 minutes, followed by Q&A based on questions received via email and any that come through live. If you do have a question you'd like to submit live, please feel free to type it in using the Q&A function within Zoom. On behalf of Arovella today, we have the CEO and Managing Director, Dr. Michael Baker. Without any further ado, I'll hand it over to Michael. Please go ahead.
Wonderful. Thanks, Matt. And thank you, everybody, for taking the time to dial in. We're very excited today to talk about the license agreement between Arovella and Sparks. And as you've already pointed out, it's for a novel monoclonal antibody sequence that targets something called Claudin 18.2 and for the use of that specifically in cell therapy. So why we're excited by it is the recent literature and data suggests that Claudin 18.2 is a very promising target for cancer therapeutics. So we're delighted to have it in our arsenal. And of course, it helps us to expand nicely because this is a target that's expressed on a range of solid tumours like gastric cancer, pancreatic cancer, and some others. So really exciting to start moving even deeper into the area of solid tumours. And without further ado, I'll go through the technology and a little bit about the license agreement. I will be making forward-looking statements, so please do take note of the disclaimer. And just very quickly, just to reiterate, many people may know it already, but just in case for those who may have joined us for the first time. So we are developing a cell therapy platform, which cell therapy is clearly going to be an important pillar for cancer therapeutics moving forward. Our cell therapy is unique. It is called our invariant natural killer T, or iNKT cell platform for short. Now, we are using that to target blood cancers initially with our lead product, LO101, that we're intending to take to clinic next year. And as I've just mentioned, we are branching into solid tumours through a range of strategies, which I'll touch on in a moment. So for this particular technology, as I said, it's a novel targeting agent called a monoclonal antibody that targets Claudin 18.2. And what we'll do is take that monoclonal antibody sequence, and we use genetic engineering so that we can have it in what's called a chimeric antigen receptor format, or CAR, and integrate that into our iNKT cell platform. Now, Claudin 18.2, as I alluded to, it is a validated target. It's generating quite a lot of interest in research articles and so forth. And there are a number of products now under development in clinical trials, and one that's going or set to be approved by the FDA in January 2024, so only in a few months away, which is exciting. And as I said, it is expressed, or when I say expressed, it's on the surface and accessible on a range of different cancer types like gastric cancer and pancreatic cancer. Now, the technology specifically for us, it's, as I said, it's a monoclonal antibody that's been developed by Sparks, and we use the sequence of that. But it has done a rigorous amount of work in terms of safety, specificity, toxicology, and efficacy, including some studies in non-human primate models. And that work has all gone into an IND submission. So it actually has an active IND through the FDA to start phase 1. In terms of the commercials, it is quite an attractive deal. We don't have any cash component up front for the license agreement. The upfront payments only in equity, which is advantageous for Arovella. And we're expecting modest expenditure for the first milestones of the project, which is great. And the remainder of the milestones, which are industry standard for a technology like this, are payable in both cash and equity. Now, this is also quite important. The technology already has very robust intellectual property surrounding it, with composition of matter claims, which is, again, what we were hoping for. It's got long patent life, and the patent is already granted in the US and has been filed in other major territories. So I'd like to talk more about the strategy we've introduced over the last few months about using our iNKT cells to target solid tumours. It is something we're very interested in and focused on because of the role that we think iNKT cells specifically can play in the treatment of solid tumours, and also the fact that 90% of newly diagnosed cancer cases are actually some form of solid tumour. So still a huge impact societally, And we know that there's a lot of work to be done to help cancer patients with these particular cancer types. And in terms of our strategies, we've got a three-pronged attack. The first, which is clearly the focus of the talk today, which is to identify and license novel targeting technologies. And we think this is something that's very important, and it's something we're continuing to pursue in the background in addition to what we're discussing today. We're also looking at ways to armor or strengthen the iNKT cells, and this is essentially to either help their persistence in the setting of cancer treatment and therefore enhance their activity. So for that strategy, we've got an option agreement with the University of North Carolina for a cytokine technology. And we're also looking to continue to create unique partnerships like the one we have between Arovella and Imugene, where we're combining both platforms, where we think there's advantages to tackling solid tumours. And just to go through this in a little bit of detail, just so it's clear, so we have seen phenomenal results for cell therapies in blood cancers. And just thinking about that from a physical perspective, a blood cancer is something that's free floating in solution. So the cancerous cells are a little bit more accessible. So it's easier for the cell therapies to find them, recognize them, and kill them, and promote their own expansion so they can continue to do the same thing. When we think about a solid tumor, they are more difficult to treat with cell therapies. I think most people probably are aware of it, but a solid tumor forms exactly that. It's a solid mass, and that can be quite difficult to penetrate. So we often talk about it's difficult for the immune cells to actually get access to the tumor. We know that there's fewer antigens that are specific. So that means they're not on healthy tissues, and uniform, which means that they're on the majority of the cancer cells of that particular cancer type. And the tumor microenvironment itself has cells like myeloid-derived suppressor cells and tumor-associated macrophages, which are part of our normal immune cell complement, but they actually turn and become pro-cancer. So they actually support cancer cell growth. So we believe that iNKT cells specifically, just based off the properties that they have, they actually are well positioned to have an impact in the solid tumor space. So a number of advantages that we see are that they actually naturally home to tissues, and they have been shown in clinic in humans to infiltrate tumors, which is going to be important for them to have robust activity. And in addition to that, the cells that I mentioned a moment ago, the pro-cancer cell growth cells, such as tumor-associated macrophages and myeloid-derived suppressor cells, iNKT cells have been shown to block or kill those particular immune cells as well. And in addition to that, through what we call signaling pathways, iNKT cells can actually help to recruit host immune cell components like T cells and NK cells, which can assist in then eliminating the cancer cells that are present. So in terms of our strategy number one, which is to find new CARs for novel targets, I'll quickly touch on our manufacturing process just to give everybody a refresher. But the way it works is instead of taking material from the patient, we collect blood from a healthy donor. We isolate the iNKT cells specifically from that donor, and then we engineer them genetically so they actually produce the chimeric antigen receptor, or CAR. And remember, I like to think about that as guided missiles. So they actually direct the iNKT cells towards the cancer cells. And once that happens, the iNKT cells trigger their destruction. So then we expand them. So that means we grow them up into large numbers, into the billions, before ultimately vialing and freezing the cells. So when we talk about new targets, what we're looking to do is find suitable CARs that we can integrate into our manufacturing process. So when all the work that we've done for our lead product, ELA-101, which is setting the stage for our manufacturing, we can leverage what we've done previously, which is advantageous. All we need to do is create what we call a new lentiviral vector, which is how we actually get those chimeric antigen receptors into the cells. Now, I'd just like to talk more about Claudin 18.2 and why we think it is a promising solid tumor target or tumor-associated antigen. Big point first up is that it has been, and what the word we use is overexpressed. And when I say that, it means that we've got the tumor cells, and overexpressed means it's produced and sticking out onto the surface of those cancer cells so that it is accessible. So a range of gastric cancer types have been shown to have that on its surface. Pancreatic cancer is another very high unmet need cancer type. And also subsets of things like ovarian cancer and even lung cancer have demonstrated Claudin 18.2 expression or availability on their surface. So in addition to that, the fact that it's there, it has been validated as I said, with the first monoclonal antibody expected to be approved in the next few months. And ignoring all the other cancer types for the time being here, just the gastric cancer market alone is expected to reach 10.7 billion by 2031. So it is a large target market and a large commercial opportunity on top of the important impact that will have for cancer patients globally. So just touching on that a little bit further, the target itself. So as I said, it is validated. And why we think it's such an exciting target is, firstly, it's not actually expressed or found on most healthy tissues and largely limited to the lining, or what we call mucosal membrane epithelial cells, or the lining of the GI tract. And when it's in those cells, it actually gets hidden or sequestered between those cells. So this is what we'd have the lining here on the outside, which actually serves like a barrier. And then we've got Claudin 18.2, which actually sits between those cells. So when we've got therapies like a monoclonal antibody or a cell therapy, they just can't penetrate to get access to it. So even if it's a healthy tissue, it's not able to have a negative effect in that setting. Now, what happens when we have a cancer that actually grows, it protrudes through that barrier, and essentially the cells undergo a range of changes at that point, but they almost, what we could say, they lose their sense of direction and they start growing in funny ways, but they actually end up producing the Claudin 18.2. And that's when, in green here, it gets pushed out onto the surface, so that when we have the cell therapy given to patients, it will be accessible, and the CAR that we're designing will be able to find that target and destroy the tumor cells. So when we talk about the major targets of Claudin 18.2, there are two major cancer types that we think of major interest. The first is gastric cancer. It still continues to be an enormous problem, with more than 1 million cases globally diagnosed per annum, approximately 770,000 deaths per annum. And depending on the stage of the disease, whether it's regional, so still locally advanced, or distant, which may mean it's metastasized, the 5-year survival rates are quite poor, somewhere between 6 to 33%, which means after 5 years, that's how many patients will survive. And it still continues to be the 4th largest cause of cancer death globally. So for pancreatic cancer, it is a lower total population of cancer cases diagnosed globally, which is about 500,000. But what you can see here is actually it's almost the same number of deaths, because it's still such a poorly met cancer type. And again, for the regional and distant 5-year survival rates, it's in the range of 3 to 15%. So still a really difficult to treat cancer and high unmet need. And essentially at this point, there's no effective standard of care treatments for pancreatic cancer that's diagnosed at late stage. So I'd like to introduce the monoclonal antibody that I mentioned that will be approved for gastric cancer in January 2024. The name of that monoclonal antibody is zolbetuximab, and it has completed now 2 phase 3 studies, and that's the data that's going to be used to demonstrate efficacy over chemotherapy alone, and will be likely approved in January 2024. It has already received a designation from the FDA called priority review, which demonstrates that this is, that's a pathway for major advances, or for where no adequate therapy exists. So that again demonstrates the high unmet need of this particular cancer type. Now, for that data, it was a 2.4-month increase in overall survival relative to the standard of care. So that's it's an important increase in survival, but through that, it's still expected to have peak sales of between 600 million and 1.3 billion for that extension of lifespan. Again, demonstrating how important and urgent new therapies are for this particular cancer type. So it's interesting, actually, Astellas acquired another company called Ganymed Pharmaceuticals in 2016, and I think it was zolbetuximab that was the focus of that acquisition. That was at phase 2, and the upfront payment for that was 422 million euros, with expected milestones of about 680 million euros. So again, some pretty interesting commercial developments for the Claudin 18.2 target. So essentially, just going back to the license between Arovella and Sparks, as I said, it is an exclusive global license with Sparks to use their patented, and the monoclonal antibody from Sparks is referred to as SPX101. So it's to use that sequence specifically to develop cell therapies. As I mentioned, the upfront fee for Arovella is payable solely in equity, so there are no cash payments up front, and modest short-term capital requirements to reach some important go, no-go decisions. In addition to that, there are industry standard milestone payments that will be made in cash and equity out to approval of the product by the FDA and other territories. So in essence, what we're doing is we're taking the SBX-101 monoclonal antibody sequence. We're not using the monoclonal antibody itself. We're using the sequence. We use that to integrate into our iNKT cell platform. And then we're going to use the combination of the two to target solid tumours. So we do believe cancers like gastric cancer and pancreatic cancer are high priority targets, especially given, we're seeing products that might be approved after survival increases of 2.5 months. We do see that there's room for better performing products. So it is exciting for Arovella as it does add a new, really important solid tumour program to our pipeline. And I think nicely for us is it does leverage the progress that we have made for ALA-101 in terms of the manufacturing. And just to reiterate, SPX1 evidence through the fact that they do have an open IND with the FDA to start phase 1, it has an excellent safety and efficacy profile to support the fact that it has that pre-IND accepted. And just to be clear, this will be the first off-the-shelf CAR iNKT cell product that is being developed to target Claudin 18.2. Now, just some quick information about SPX-101. So this is now looking at the monoclonal antibody version of the drug that was developed by Sparks. And what they've actually done here is demonstrated some important functional attributes of their SPX-101 monoclonal antibody. And they've actually compared that to a version of zolbetuximab that they've manufactured internally. And so what we saw was it has what we call 30-fold higher affinity. So it has the 30-fold times better ability to target and bind to Claudin 18.2. And they've seen superior efficacy of the SPX-101 in animal models where they also express or have Claudin 18.2 on their surface. So that's what we're looking at on the graph. On the vertical axis, we have the tumor volume. So the lower the curve, the better. And we're looking at here in terms of number of days. So zolbetuximab is in the pink line, salt water, which is placebo in the black line, and SPX-101 is in the blue line. So clearly there's a better performance in terms of tumor control. And as I've mentioned, Sparks has an open IND with the FDA to commence a phase 1 in patients with gastric cancer. So how do we actually do what we want to do with this technology? I'll go through that briefly here in this cartoon series. We have the monoclonal antibody is the whole picture here on the left, SPX-101. We're interested in this part that's in the green box. That is what we use to target the Claudin 18.2, or that's the guiding part of the missile, let's say. So we take that, again, this requires some genetic engineering, but we make a chimeric antigen receptor, which has the targeting region or the guiding region on the end there. And then we use genetic engineering to integrate that into our iNKT cells. So as I said, this will be the first off-the-shelf CAR iNKT cell product targeting Claudin 18.2. We do have the benefit of seeing some data from an autologous or non-off-the-shelf Claudin 18.2 CAR T product. And that's already started to generate promising data, which gives us, again, good signals for working with iNKT cells because of the additional benefits we think the iNKT cells can present for solid tumours. So in terms of the manufacturing, as I outlined before, this is exactly what we're going to be doing now. We take the Claudin 18.2 CAR. We'll use our manufacturing process that we've already got and continuing to develop. We basically get or buy or purchase or build a new lentivirus that then goes into the iNKT cells so they can express the CAR. We get the Claudin 18.2 CAR iNKT cells manufactured. We expand them or grow them up into billions, and that's when they're ready and available for use. Now, I mentioned the patents earlier. As I said, this has got very long life out until 2038, which is excellent. The claims for this are composition of matter, which is excellent. That's the strongest IP we can get. and it's already granted in the US, which is excellent. So we've got pending applications already on foot in Europe, China, Japan, and South Korea. So targeting most major markets, which is excellent. And I'll just finish by talking quickly about our second strategy. As I said, this is important that we continue to build on a range of strategies for solid tumours. So as I said, this is the armouring approach, where we're looking at the cytokine technology. And what we've done is entered into an option agreement with the University of North Carolina, and we're adding a cytokine technology to increase the persistence, or how long the iNKT cells actually last in the body, so that we actually increase the anti-tumor activity. So when we've used that, or we see that in animal data, we do see that the iNKT cells expand more and survive longer. So there's approximately 10 times more cells or CAR iNKT cells at the 4-week mark versus iNKT cells that do not have that cytokine technology. And not surprisingly, we also see what we expected, which is superior anti-tumor activity, with 75% of mice with that technology alive at 61 days, and mice without that technology all dying within 49 days. And I should point out, this is a separate solid tumor model that they've used for a different CAR. But you can see clearly there's opportunities for the strategies that we're employing on solid tumors to be used in parallel. The nice thing about that is, when we want to integrate something like this cytokine technology, we don't need to redo all the manufacturing work. We use the same process, and we can include the CAR and these technologies in the same lentivirus, which then helps us to express those and have them integrated into the iNKT cell platform. So in terms of our pipeline, we do have some exciting things ongoing. ALA-101, which we're using for CD19 expressing leukemias and lymphomas, looking to take that into clinic. We've got our ALA-101 onCARlytics collaboration with Imugene, which we think could be useful for a range of solid tumours. We're excited to get data out for that before the end of the year. And we've got, as we've talked about today, I'm very delighted to have the introduction of a new and validated target, Claudin 18.2, which we think can be useful for a range of solid tumour types. And of course, the cytokine technology, which we believe can enhance the platform across any of the solid tumours that we look to target. So in terms of next steps, it's going to be a busy 12 months and a busy start to end to 2023 and start to 2024. So major focus is to advance ALA-101 into its first in-human phase 1 clinical trial in 2024. So to do that, we're continuing to work through process optimization and preparing the process for clinical grade manufacture. We are on track to complete some of the components to CGMP levels, which is exciting, and we'll look forward to announcing those when they're completed. And in the background, we're working to finalise the plan for the phase 1, the clinical trial plan for the phase 1 study. So early next year, the plan is to continue and manufacture material for phase 1 while we're completing the necessary documentation to submit to regulators like the FDA and the TGA to start phase 1. So in terms of our preclinical stage platform or programs, for the CAR-19 iNKT cell onCARlytics collaboration, we're looking forward to getting data for that before the end of the year. And this is a point that we have now, based off today's announcement, we have made progress. We have in-licensed a new CAR to add to the platform. And for that, for the remainder of this year and beginning of next year, looking forward to initiating proof of concept testing to demonstrate the potential of this particular CAR in the iNKT cell platform. And for the cytokine technology, we're continuing to work through that, and pending our due diligence, we'd look to in-license that into the company as well. So I'll just summarize there. So as I said, we've got a very exciting cell therapy platform. It's a novel allogeneic or off-the-shelf iNKT cell platform, which we're using to create therapies for the treatment of blood cancers and solid tumours. We've demonstrated that iNKT cells, CAR iNKT cells, have multiple anti-cancer properties. For blood cancers or other solid tumours that do produce CD1D, we see that they do become dual targeting. For the off-the-shelf component, we see that that's a really important step forward for the cell therapy sector, because the improved logistics that will arise from that would alleviate a large bottleneck, which is the manufacturing as it stands currently for the therapy for every single individual that needs to take it. So excited to take our lead product, ALA-101, continue to advance that, looking forward to taking that into clinical trials and expecting that to happen in 2024. And we have talked about it earlier, but just reiterating the fact that iNKT cells do have properties that are unique to iNKT cells that we think will help them target solid tumours, like the fact that they already infiltrate tumours, they home to tissues, they start to influence those pro-cancer cells in the tumour microenvironment, and they also help to bring the host's immune cells like T cells, NK cells, to join in the eradication of the tumour cells. And delighted that we've expanded our pipeline through the addition of the Claudin 18.2 CAR, and looking forward to getting that up and running and starting to develop that to target gastric cancer and test it against pancreatic cancer as well. So with that, I'd just like to say thanks everybody for those that have dialed in. It's really great to have you listening in. We're very excited by this new development today, and I'd certainly be delighted to answer any questions.
Thanks, Michael. We'll move on to the Q&A, as you've mentioned. The questions are based on those sent through via email and any using the Q&A function within Zoom. So as a reminder, if anyone in the audience does have a question you'd like to submit, please feel free to use that Q&A function and type it in and submit it on your screen. The first question in the queue we have is, what are the next steps in terms of development of this technology, Michael?
So good question. The first thing we need to do is, based on that diagram I showed earlier, is take this from the monoclonal antibody sequence and put it into the CAR format. That we're not expecting to take too long. We can design that and start producing that quite quickly. And then we get it into the iNKT cells and demonstrate that it has the properties that we want in CAR format, and then start doing some of the in vitro and in vivo studies in animals. So that we don't expect to be a terribly capital intensive program, but we'll get a lot of good data. And of course, if that data's at key milestones, we'll be looking to announce that as well.
Thank you. The next question is around competition in this space and how much of it is there.
Look, competition is always something we're mindful of, and I think for a target like this, it's now at the tipping point where I think it could become quite competitive. And delighted that we're able to enter it in a way where we've got a monoclonal antibody that's already done all the work that we would like it to have done to get the approval from the FDA to start a phase 1 clinical trial. So that's quite a lot of information that we don't now need to go and replicate, which is phenomenal. So looking through the literature, there are several monoclonal antibodies being developed, like the one from Astellas, which is predicted to have sales of somewhere between 600 million to 1.2 billion. A range of other therapeutics like antibody drug conjugates, bispecific antibodies, but there are only a handful of CAR T products. So there's not much going on that front, and there are no CAR iNKT cell products. So we again sit as now the only company in the world that's developing a CAR iNKT cell product targeting Claudin 18.2. And again, because of that data from Sparks, we feel like that we've got a pretty good or some advantages moving forward. And we can just focus very quickly on the efficacy data, which is great.
Thank you. And another question is, do you expect any difficulty in recruiting for a trial of such technology?
Yeah, good question. And I think largely off the patient numbers for diseases like this, as I said, for the gastric cancer market or global patient numbers, it's more than 1 million cases. Sorry, yeah, exactly, more than 1 million cases globally per annum. So in terms of patient numbers, we expect that there will be quite a lot. When we look at the CAR T trial from Carson with their phase 1, they had a phase 1 study over 2 years, and they were able to infuse 49 patients within that time frame. So I think the high unmet need, the lack of effective therapeutic options for these patients, and then the concept that we could have an off-the-shelf product available for testing, I think that could be quite compelling. So quite a number of patients in that CAR T study, they actually fell out of the eligibility criteria to take the therapy, because back on that CAR T scheme, it's about 4 to 6 weeks for the therapy to be manufactured, and patients with aggressive disease progress, and then they no longer fall within the eligibility criteria to actually receive the therapy. So we don't expect to have too many problems recruiting.
Thank you. The general question that's come through is, how's the company looking to fund up and coming trials of your technologies?
Yeah, absolutely. And again, I think that's quite a nice way the deal was structured, right? We've got no upfront fees that we need to pay in cash, which is helpful. And we think to reach the first inflection point where we demonstrate the potential, it's going to be less than $500,000 to complete those studies. So that can all be funded out of our existing cash reserves. If the data looks good and we're willing to take that forward, then of course, and depending on the next and upcoming milestones, we may look to do an additional capital raise. But for the early stage work, I think we're pretty well funded, which is good.
Very good. And another question that's come through, could this Claudin 18.2 CAR be combined with Imugene CF33 in the same way that you're combining ALA-101 with CF33?
It's an excellent question. I'd like to meet the person that's asked that. So look, long story short is yes, you can do all these things, but what you need to do is have now a CAR that targets Claudin 18.2 and a CAR that's targeting CD19, and then you would use the virus to produce or to infect gastric cancer cells. So those cells would now be in a position where they actually could be producing Claudin 18.2 on their surface and CD19. So then there's a possibility that you've got a dual targeting CAR, you're covering more of the tumor cells with CD19. So it's something we haven't discussed, but it's certainly a possibility scientifically.
Thank you. And another one that's just come through, do you expect these new applications to be used in humanitarian cases beyond clinical trials?
I'm not sure that I understand the question fully. Can you just say that again, Matt? So do we expect the...
Read word for word here. Do you expect these new applications to be used in humanitarian cases beyond clinical trials?
Yeah, I see. Look, it's always a possibility. I think for things like pancreatic cancer, where the need is so dire, and I assume what this is referring to is special access schemes where you'll get the consent of a principal investigator to actually dose people off label. And look, it's still very early to determine if we go down the pathway for something like that. But notwithstanding, you will still need to have product manufactured at suitable levels that you would typically take into a clinical trial. But certainly, if we have that available and there's scope to do something like that, we could explore it.
Thank you. And another question, how significant is the achievement of manufacturing?
Yeah, it's very, very significant, actually. So for cell therapies, it's often mentioned that the process for manufacturing becomes the product. So that's why we're delighted to have made good steps already in terms of getting high amounts of CAR into the iNKT cells. And again, what we've shown previously and what we presented at AACR, that we can grow those cells more than 5,000 times and still have good activity in vivo in animal models. So we're making good steps, and it's a key focus for us to get that process locked down, and then we start to scale up so we're producing material. And I think it's a critical milestone, not just for that program, but of course for things like the Claudin 18.2 CAR, because then we just feed them into the same manufacturing process.
Another question that's come through, what is the possibility of getting priority review status or orphan drug designation from the US FDA and in Europe?
Yeah, it's a little bit different between the two territories, but essentially what you can have when you have different schemes like that is you may get faster approval for those therapeutics, and you can also get increases in exclusivity post-commercialization of those therapies. So I think why that's important, it just demonstrates, for the priority review, how unmet the medical need is, which is important. And then on top of that, you can get those additional benefits of faster path to market and some exclusivity around blocking entrance. So again, some commercial advantages as well. I think for the priority review as well, you may get a voucher that can be used for other programs, and that sometimes they're sold in the order of $100 to $200 million as well.
Thank you, Michael. I've got no further questions, so I'll just throw it back to you to provide a closing comment.
Thanks, Matt. Yeah, just like to reiterate, this is a wonderful step forward for the company. We're incredibly excited by the target and certainly what we think it could do in combination with iNKT cells. But again, just reiterating, big thank you to everybody that's dialed in today. We appreciate you listening in. Thank you.
Thanks, Michael. Thanks, everyone, for joining.